I attach it to me?
Yes, you can attach it to your pants, I think is the easiest thing to do.
Fit it, at least it won't come off.
You know what, push, it's on, so let's just.
Blow this is.
You guys, please make sure to sign in if you haven't yet. It's here and it'll get passed around. Thanks. I was just going to say that.
See?
All right, we're going to go ahead and get started. I'm Kira Larson. If I haven't met you guys, I'm the Education Program Manager in the Stanford Center for Clinical Research. I will introduce Kathy in just a minute, but wanted to make you aware of the next SCCR Science Series on June 22nd. Same situation with a flyer. We'll go around. If you didn't get an email from me with the information, let me know and I can add you to that listserv because it's my own personal listserv. But it's June 22nd and it's on, I just was talking about it, it's on medical terminology and common medical acronyms. And we have invited one of the internal medicine residents to come and give that lecture. So again, sign up the same way, but let me know if you aren't on the email list and you didn't receive it. Okay, so this is Kathy's second time back at Stanford, and we are excited to have her back. Kathy obtained her Doctor of Pharmacy from the University of Michigan and is a California board-certified pharmacotherapy specialist. She is a Health Sciences Associate Professor of Clinical Pharmacology at UCSF School of Pharmacy. She also currently practices as a clinical pharmacist at UCSF Mission Bay Benioff Children's Hospital in San Francisco. Her primary focus is drug safety and surveillance and medical group quality assurance. She has practiced as a pediatric hospital pharmacist, a drug education coordinator, and as a clinical operations manager. Her research interests include adverse drug reaction monitoring and pharmacovigilance, medical group quality assurance, pediatric pain management, and hospital pharmacy practice. She's an expert educator in the field of pharmacology. So again, please welcome her back to Stanford.
Thank you. Am I on? Can you hear me? Oh, good. Well, now let's look around the room and look at all the ladies in science. Wow, I'm old, so this is great. This is great. Thank you, everyone, for coming. I like to have a very interactive talk if possible. You're all learned. And you may well be doing research in an area or have a background in an area. Just raise your hand and join in. So I hope today to give you information that will help you in your research endeavors, especially and obviously if it involves drug therapy, which as a pharmacist we think everything involves drug therapy. So I'm going to talk about kinetics, dynamics, and genomics. So my new hobby or interest, if you will, is PharmacoGENOMICS, which I truly believe is going to be a pivotal change in medical science, much like germ theory or vaccination. And soon we'll have answers to diseases that we never knew the cause of. We have so much more that will help us personalize drug therapy. So it's an honor to be speaking with you today. So the goals of this lecture are to help you differentiate between these sciences or bodies of knowledge we call pharmacokinetics, pharmacodynamics, and pharmacogenomics, and mainly how they are overlapping. We're going to review the mechanisms of drug absorption, distribution, metabolism, and elimination, which is the heart and soul of dosing of many medications that pharmacists have, for the most part, taken ownership of. I hope you'll gain an awareness about how patient-specific gene variation may influence these processes. And this is science in its infancy, it will be very likely that the pharmacist will have a big role in looking at people's genetic profile, if you will, or their genome, and where it makes sense, adjusting drug therapy based on their specific genetics. So we hear about these terms, personalized medicine. Well, nothing's more personalized than your individual genetic makeup. And then I will direct you towards resources where you can look up information. So what is have you heard this term personalized medicine? Okay, what does it mean to you? Anybody?
Personalized medicine is the concept of like studying your basic genetics and what therapy defining the treatment.
Yes, so yes, matching the therapy we give you with your individual genetics. It used to mean looking at things like your renal function, and in reference to drug therapy, and adjusting the dosing of drugs, and making that part of your personal therapy. So I started my practice in pediatric hospitals, first 6 years, and I remember when I was thinking about getting a job at an adult hospital, I remember thinking, oh no, I don't know if I can treat adults. I've only worked in pediatrics. They go, adults all get the same thing. Everybody gets 25 milligrams of atenolol. Everyone gets, 10 milligrams of hydralazine. They all get the same thing. And I'm like, okay. And indeed, it made it really easy. We're children. We always have personalized their drug therapy, mainly based on weight. But each child was dosed. This is now changing. And so think of how we vary in size, in shape, in age. And we'll talk a little bit about that. So this whole idea of personalizing or matching the therapy better to the human being is going to be very important. So have you heard of the Human Genome Project? Which is just fascinating. The thing I love most about it, and I've read about it, I'm certainly no expert, is that the reason it was completed so soon, and what that project was, is a worldwide effort to map out every gene that a human being possesses. It keeps changing, but there are about 22,000 genes. Sometimes they say 30,000. I guess they'll find more. The reason it was successful is that it was a group effort, a worldwide university, medical researchers, everybody pooled their brain power. And I love the spirit of that. I just think that's so great. And that is why it completed so soon. I think the original completion goal was something like 2010 or 2015. So I think that was a great thing. So the goal was to sequence base pairs, and there's only four nucleotide bases that make up the DNA molecule. And from those four letters, if you will, it spells out every protein that goes into making a human being, and many other things as well. So, as I said, it allows a much better way to fit medication dosing and selection to each person's unique physiology, and it's definitely an exciting time to be in healthcare and to be a healthcare researcher. So it's good to think about drugs as molecules. So in pharmacology and medicinal chemistry, you think more about drugs as molecules because it's the molecule that has effect in the body, that the body deals with. And this is aspirin, old aspirin from early 1800s. It was synthesized. So the original salicylic acid was extracted from willow bark. And this was one of the first drugs that was synthesized. So aspirin works because the acetyl group cleaves off and binds to cyclooxygenase, or COX-1 and COX-2. So you've heard about COX-2 inhibitors like Celebrex. They work because those molecules as well, inhibit cyclooxygenase, which is an enzyme, and enzymes are proteins, or at least contain proteins. That's good to know. So the pharmacological effect of aspirin is many. If this acetylates cyclooxygenase, an enzyme, that enzyme is responsible for converting precursors to prostaglandins. And prostaglandins do all kinds of things in your body, but they mediate pain. So if you lessen the amount of prostaglandin, you relieve pain. You also do a few things that we don't really like you lessen the ability of the stomach to produce its mucus coating, but I call that, instead of side effects, I call that alternate pharmacology of drugs, the dark side of drugs. So it's good when you're thinking about pharmacodynamics and pharmacokinetics to remember that we're dealing with drug molecules. Okay, so let's talk a little bit about the differences in these terms. So pharmacodynamics, when I went to school, we would call that pharmacology. It's how the amount or the concentration of drug at its site of action affects its physiological action and toxicity, or the mechanism of action, how the drug works, what it's doing in the body, and mainly what it's doing to the body. So pharmacodynamics is how the drug molecule acts on the body. Now, of course, drugs do all kinds of things, but a lot of them plug into receptors. So that's that little guy there. That's supposed to be a plug. So there are receptors on cells. There's receptors on bacteria. There are protein binding sites. Typically, these contain protein. That's something to think about. Not every drug plugs into receptor. We give sodium bicarb. For the most part, it's changing pH, and there are other things going on, but a lot of them do. So pharmacodynamics is how the drug molecule acts on the body. Pharmacokinetics is not the opposite, but it's how the body acts on the drug molecule. So anything you ingest or byproducts need to be eliminated from your body, including drugs. So pharmacokinetics is more the study of what the body does to that molecule and how it gets rid of that molecule, or how it converts the molecule. So it's a science of quantifying the amount of drug throughout the body, from absorption to elimination. So as a pharmacy student, you take courses in pharmacokinetics, and it tends to be a lot of formulas that predict drug doses. We figure out, or we've always personalized the dosing for many medications by figuring out how much is in the patient and how long it lasts, and that patient's ability to get rid of the drug. And we'll talk just a little bit about some of the terms, because you will come across them, things like half-life and clearance. You'll come across them if you're doing any studies involving medications. So once again, kinetics is how the body acts on the drug molecule. So now we have genomics, and we talked a lot about that. It's a science that relates specific variation in genes with differences in drug response. We're not going to talk too much about that today. Differences in an individual's propensity for developing certain drug side effects. So certain side effects, we always say they're idiosyncratic. They just happen. We don't know why. Well, pharmacogenomics is really going to shed a lot of light on this. We will, and hopefully the best thing that'll come out of that is we'll be able to test people and say, oh no, you shouldn't have Tegretol because you are at a higher risk for having these terrible skin toxicities. So the potential for these kind of associations is going to be wonderful for people. And what we're going to talk about a lot here is the variation in the rate and extent of drug metabolism. All right. So let's start at the beginning. You got to get the drug in the body. Anyone do any kind of research that involves new dosage forms or any of that going on here? It's actually a, oh yeah, go ahead. It's a what now? Botanical extract. Oh, and you're looking at how, what it the levels in the body, or?
No, we are actually treating devices with a botanical enzyme. So we are testing the safety and tolerability.
Awesome, awesome. So, yeah, so some of this hopefully will apply. And is it taken orally?
It's oral.
Okay, so most things are taken orally, but we have all kinds of ways of getting drugs into the body. So percutaneous is through the skin, patches, which really, patches aren't, things like nitroglycerin paste, that's an old product. But patches haven't been around all that long, maybe 25 years. It's not for every drug. There again, the chemistry of the drug has to lend itself for being soluble enough in a fat layer as well as an aqueous layer to be able to absorb through the skin. So things like fentanyl patches, that works really pretty well. Estrogen spray can go through the skin. We can also go through mucous membranes. So mucous membranes are nice and thin and they're highly vascularized. So certain things can go under the tongue or buccally, intravaginally, rectally, sublingually, nasally. Anyone have allergies and have their Flonase out? That's you're absorbing that into your circulation as well as topically. Let's see, injection, of course. But there's other types of injection than just going into a vein. You can go into the subcutaneous. It's not really circulation, but the subcutaneous layer, and it diffuses and ends up being absorbed into the blood vessels, intrathecally into the spinal column or to the CNS. Intramuscular, we do use that a lot for drugs. And intraosseous or osseous, which sounds horrible. There are special devices that they actually can use in ERs that'll go right into the femur. And it's a wonderful avenue to give a lot of fluid, especially to children who have soft bones. And believe it or not, it's not as painful or awful as it sounds horrible, but it's actually not. And it's a great way to give a baby lots of fluid. I know. And they do it for adults too sometimes. Inhalation. So all kinds of drugs have lung absorption. Anyone remember the inhaled insulin? Yeah. Yeah.
No, I didn't use it personally. It looked like a bomb. So
Yeah.
it did not sell it.
Yes, and that was a big downfall. Yeah. So it actually worked, but the device, was like this big old thing. So the selling point was, people don't have to inject themselves at the dinner table or whatever they can, but it, yeah. And I think they related it to an increase in asthma or, was it asthma?
Stuff that occurred.
Yeah. Nonetheless, I applaud the effort because they have been trying to get insulin into the body in another way for forever. Why do you think you can't give it orally?
Actually,
it doesn't absorb. Why? It's a chemical. It's a big protein. What happens when you eat protein? You digest it, yeah. So yeah, it's been a real bone of contention. I think the inhaled insulin is making a full circle and coming back, and I think they're still working on some oral way to do it, which would be great. So no matter how you try to get a drug in, the amount that gets in, the percent that gets in from the dose is referred to as bioavailability. So if you look in a standard drug reference, you will see information on oral bioavailability, rectal bioavailability, how much ends up in the bloodstream for various reasons. Full absorption. Yeah, sometimes you have to wait. Sometimes it takes a little bit to absorb. So for an IV, pretty much yes, because you're going right in, and we're measuring serum blood levels for the most part, though I think they can measure other deposits, and we'll talk about that. Oral absorption takes time, so they typically will, so a new drug, like your herbal product, someone will likely do bioavailability testing. They'll give a known dose and take serum samples over time to find out when it peaks and, yeah, how long it takes to absorb. Yeah, yeah. Okay. So absorption, oral absorption has a few considerations. First of all, most of drug absorption happens in the small intestine, past the stomach. So you got to get through that stomach acid and into the intestine. You got to have a product that dissolves. So when FDA approves a generic, and you'll hear that it's bioequivalent, a lot of that means that the product dissolves and gets into the body, because you can make a pill that contains, 5 milligrams of drug X, but if it also doesn't dissolve, yeah. Yes, but no one's sure if that enteric coating really does a thing. But yes, you got it. It's to try to, well, it's not so much to protect the drug from breakdown. It's more to try and spare the stomach lining from direct abrasion. And does it work? Some studies say no, but we do it anyways. Yeah, we think it works. Yeah. So these things are a consideration. Does your dosage form dissolve? What's the stability of the drug in the GI tract? Some drugs actually bind to each other in the gut, and we can get drug interactions that happen and interfere with absorption. That's one kind of drug interaction. So, yeah, go ahead. Yeah.
So I have an incredibly robust sensitivity to vitamins.
Yeah.
I get physically nauseous when I eat them, even if I have eaten food. Why is that?
Yeah. Why is that?
Anything about me specifically?
No, that's a good.
Like vitamin C, multivitamins.
And they upset a lot of people's stomachs.
Like, it's as though it's almost, I would think, breaking down too quickly in your stomach before then it can get here. It just may be an allergy or something.
It's not an allergy. It's very common that they make, actually there are a lot of drugs that make people nauseous. That doesn't necessarily mean it's a thing like aspirin where it's caustic to the stomach. That's a darn good question. Why do vitamins make you nauseous? Okay, I will get back to you on that. Does someone know really? Because it's very common.
I feel like it's almost weird.
Yeah.
That my stomach is so upset.
It also might be, there might be some vitamins kick up stomach acid. It could be some of that. I'm going to look that up because now I want to know. Because that's pretty common. Yeah. But it's not dangerous. It's just uncomfortable. Okay. All right. Good question. Yeah.
One of the drugs that binds with each other.
Yeah. Some of the antibiotics, and so levofloxacin will bind to calcium. Tetracyclines, doxycyclines will bind to calcium. So that's why you'll see those our cute little stickers, do not take with dairy. Excuse me? Yes, that's coming up. Yeah. Oh yeah, that's coming up. Yeah, the grapefruit interaction. It's a little different. The mechanism's a little different there, but there are many drugs that you should. Some drugs, like probably the one in widest use is levothyroxine. So thyroid. You always take that on an empty stomach because you won't absorb it as reliably alendronate or the bisphosphonates to harden your bones, if you will. Always empty stomach drugs. There aren't really all that many, but calcium, you're right, the positive cations like to bind to drugs and prevent their absorption. So I have to show these slides because one of my students made these actually quite a few years ago. Drug absorption through the duodenum into the bloodstream. Isn't that fun? And then distribution, we're going to talk about that next, to other parts of the body. All right, so here we go, drug interactions at absorption. Oh yeah, so things like Metamucil and the fiber laxatives like to bind drugs. So they're kind of the rule in pharmacy, and who knows if people can actually manage this. I tell you, some of the things we tell people to do is to separate. So if I have to take my levothyroxine, I'm going to do that an hour before I take my Metamucil. Well, in that case, or 2 hours after. So we like to use those windows. Is that really valid? Well, we're not so sure. Calcium salts and iron, they're positive cations, they bind to drugs, and we talked about, yeah, Synthroid or levothyroxine. So there we go, this is her second slide. There's her iron and her cheese, her calcium, isn't that cute? All right. So now we're going to talk about grapefruit juice, because this interaction is a little bit different. These things are found serendipitously too, and that happens a lot in science. So there was a calcium channel blocker, felodipine, that's actually hardly used anymore because there's lots of other drugs, mainly amlodipine, that don't have this effect. But I think they were doing studies on felodipine, and somehow they put 2 and 2 together that if it was taken with or in conjunction with grapefruit juice, you got a huge amount of absorption. And I think it was actually when they were doing the bioavailability studies and were just happened to use grapefruit juice as the chaser. This is dangerous because it increased the absorption so much you would get double the blood level of this drug that affects heart rate and vessels and all kinds of things. So a lot of research has been done, and what happens is there's a chemical in grapefruit juice, furanocoumarins. It's also in something called a Seville orange. But I don't know, I think the Europeans like Seville oranges. I don't know how common we get anything with Seville orange in it. But what this chemical does is it binds to CYP3A4, and we're going to talk about P450 enzymes later on. But mainly these enzymes are in the liver. That's where most of drug metabolism happens. But you also have it in your GI tract. Well, the furanocoumarin gets there first, binds up the enzyme, so the enzyme can't do its first breakdown of the chemical felodipine. So there are lots of drugs that they'll have warnings about. The statins. So there's, oh, don't take it with grapefruit. But we really haven't been good at relating the level of statin. So it increases the level of atorvastatin. Well, it might be a good thing. We don't have a good, clear picture of how the level of a statin relates to its toxicity, which is liver toxicity or muscle toxicity. Yeah, rhabdomyolysis. The occurrence of rhabdo is something like 1 in 10,000 patient years, yet the public is very aware of this and they're frightened of it. So you probably shouldn't take it with grapefruit juice. That one's a little wishy-washy. Something like felodipine, yes, you do not want to take that with grapefruit juice. Probably, I think what we end up telling people now is just don't take grapefruit juice with your medicines. Separate it. If you love grapefruit juice, it is yummy. Have it in the morning and separate it from your medications. Sometimes we'll tell people to take a certain drug with a high-fat meal. So sometimes it increases the absorption, sometimes it limits the absorption. So efavirenz, which is a antiviral for HIV, the absorption is better with a high-fat meal. So we would tell patients that they should eat it with an avocado or something that contained fat. Distribution. Okay, so now we got the drug and we've absorbed it mainly in our circulation, but really, and we like that because that's where we sample, and so we think about the levels flowing through your bloodstream, but really drugs distribute far and wide for the most part. It's different for every drug. This information is typically in a monograph or the package insert, and we'll talk about that later, how to get that information. But mainly, we want that drug at the site of action, right? So let's say I'm treating meningitis. Where do I want that drug level? Where am I concerned about that, the amount of drug? Yeah, CNS. So not every drug goes into the CNS. It has to be a certain type. Not every antibiotic does. Not every antiviral does. So your infectious disease people know that. They know which drugs to use based on the site of an infection. Pain medicines, we want to make sure that they get to the periphery and the CNS as well. Drugs bind proteins too. Now, where do you have a lot of protein? Kind of all over the place, huh? You have albumin flowing in your bloodstream too, and the level of albumin in people varies. A lot of times the elderly or chronically ill have low albumin levels. So if the drug, some of the distribution of this drug is going to be bound to albumin, that can be different. So for certain medications like Dilantin, we will measure a free and a bound amount. So this could get really complicated. The lymphatics, does anyone ever think about the lymphatic system? I kind of don't.
With cancer.
Yeah, I guess so. Yeah, I mean, it's kind of important, it's kind of ignored. But it's quite a large volume, and yeah, and we want certain drugs to perfuse that well. Bone, so treating things like osteomyelitis or the drugs that increase bone density, we want their action is the bone. Muscle, fat. So this is a challenge to all those chubby people, and there's lots of us now. That's really changed dosing and how we look at drugs, because drugs tend to permeate fat tissue, but not to the extent they do other tissues. So it's a new area of research and concern. When we have very obese people, how do we dose them? And there's formulas for that. And of course, your hair and nails, drugs will deposit. I don't think every drug does, but the illegal drugs, they know a lot about. Okay, so pharmacists spend a lot of time with volume of distribution because it's a factor we need to help us dose a drug. So it's almost a model that kind of isn't a real place in your body. It's more of a conceptual, mathematical thing. So it's the size of the total space measured in liters that a dose of a drug will fill. So vancomycin is one of our workhorse antibiotics for serious gram-positive infections, and we know a lot about it. And it's one that we personalize the dose of. So we have a formula for figuring out, in my person, we take their weight, multiply by 0.7, and that gives us the amount of volume we need to fill with drug to meet a certain level. That's volume of distribution. Drug levels in body tissue, though, don't necessarily parallel serum concentrations, but we work with what we have. So amiodarone is a great example of this. So this is an antiarrhythmic. It's somewhat in wide use. And everyone remembers it because one of its side effects can be that it can give your skin a bluish tone. I've actually never seen that. Yeah?
But I have a funny story because I had a patient on completely blue, like a blueberry.
Oh, wow.
I thought it was the drug, but it was actually blue.
Cyanotic?
No, it was blue coloring in the tube teeth.
Wow.
So it was shocking.
Oh, wow. Wow.
Because if they aspirate it.
Oh.
I don't know.
Oh, yeah, methylene, a little bit of methylene blue.
Methylene blue?
Yeah, they still do it.
Really?
Well, not routinely, but if they're looking for a leak, just because I sent it. Yeah.
I don't know if they still do it. But I will never forget amiodarone for that.
Yeah. Yeah.
I mean, I from pediatric world, I've never seen that in pediatrics.
It's tricky. I have sent it with, it's just one time, they just do it one time. I think they're looking for some kind of leaking. Does that make sense?
Well, the aspiration does. It can be hard to tell if it's secretions or.
Okay. All right. So they do it for that kind of assessment. They don't use it a lot. And yeah, you wouldn't want to give a lot of methylene blue.
Interesting.
Yeah. Well, amiodarone is an interesting drug. So it's so protein bound. It distributes out of your bloodstream and keeps binding. So at the end of the day, and it takes like a month to get people to steady state, at the end of the day, it has a massive volume of distribution. So does that kind of help you conceptualize this? So it's 148 liters per kilo. So in our 70 kilo patient, it's 10,360 liters. That, to have the concentration in the bloodstream match the total amount in the body. So those are kind of an exceptional drug. So PharmacoKINETICS, we spend a lot of time looking at plasma levels because we can get them. And there are good correlations between plasma levels and efficacy or toxicity. For certain drugs, I'd say there's about 12 to 15 drugs where we really do a lot of work with drug levels. So vancomycin, we talked about that. Cyclosporin and tacrolimus, which are anti-rejection drugs, they have a good handle on what that blood level needs to be to keep the person safe and to make sure that they're therapeutic and don't reject their organ. Dilantin, so the anticonvulsants, we do a lot of drug levels, not as often as we used to, but a quality recommendation is that if you're on an anticonvulsant, things like phenytoin, valproic acid, you should have a level at least once a year, probably more often. And then lithium. Lithium's a little tricky. Things can affect its level. That can lead to renal toxicities if it gets too high and you want it to be efficacious. So there's a window of serum levels that we want to shoot for. So I'm sure you've heard of peaks and troughs, yeah? And we talked a little bit about that, absorption, the peak, and the lowest point before the next dose is given. So let's say that this is the third dose of vancomycin. So you're not really at a steady state. So when the dose was given, is this going to work? Oh, that's right. Okay, that's all right. When the dose is given, we were at a serum level of 10 mics per ml. And then we gave our dose, and it went up. we infuse this drug over an hour, so it takes a little. So at about an hour and a half, we go to a peak of 60, and then it drops until the next dose is due at Q12 hours. That's a typical drug that has a somewhat short half-life where we give multiple doses, and that has linear kinetics. So it makes sense. You give the drug, the body metabolizes it, you give it again. And we used to, for many of these antibiotics, look at the peak and the trough, and we'd have goals for the peak and the trough. But newer theories have made the relationship between the total exposure of the drug over time. We'll look at that a little bit. So this is a patient at steady state. So you reach steady state after about 4 to 5 half-lives, where it just means the amount you're replacing equals the amount you're getting rid of, and if all goes well, there's a steady state with each dose. For the drugs that we tend to concentrate on in PharmacoKINETIcs, there we go. Our goal peak is below area of toxicity, and our minimum level occurs, we want to keep it above a certain level to keep it efficacious. This area under the curve, we use this for very few drugs, vancomycin, carboplatin, which is a chemotherapy agent, and it's simply that somebody has related outcomes to this total amount in the body. So all that's done there is, if you went through and made little rectangles and summated the total amount of drug over time, that's what's the goal of dosing is. So for carboplatin, they'll dose it at anywhere from 3 to 6 area under the curve. And we use these calculators to figure it out, but it ends up being a strange, because it's an area. It ends up being micrograms per ml per hour, or it's just a different way of looking. And so the way to think about that is total exposure of drug over time and how that relates to toxicity and efficacy. Okay, so now you've got the drug in the body, and you let it spread out and distribute. Now your body is going to get rid of it like it does with any foreign substance or byproducts of cellular biochemical reactions. So typically molecules go through chemical changes such as oxidation, reduction, and hydrolysis to make them more water soluble so they can be eliminated. Enzymes called transferases can add glucuronic acid to a drug molecule and make it more soluble. Esterases can hydrolyze drugs in the circulation and in cells. So there's lots of processes that go on. Most of true drug metabolism happens in the liver, in liver cells, but that's not absolute. So about 75% of all drugs are oxidized by this enzyme system called the Cytochrome P450 enzyme system. I'm sure if you work with drugs, it's something always considered because it can be an area where drugs interact. Okay. There's Liverland. So this is really how I think about it. So drugs are absorbed in the circulation. They pretty much dump from the duodenum circulation right into the liver. So we sometimes call it a first pass effect. They get processed, and they either form inactive molecules, or sometimes you need this process to activate a drug and to get to the pharmacologically active metabolite. Because they're more water soluble, a lot of the excretion ends up in the urine, but it can be in feces, it can be in bile. sometimes that bio will recirculate and you'll reabsorb and eliminate, so it can get very complicated. So this is kind of a hardcore way to look at this. So phase 1 reactions in the P450 enzyme system make the drug more water soluble, and so the first step, they can either convert the drug to something that's inactive or an active metabolite. Sometimes it's the metabolite that causes side effects and body injury. Then other phase 2 reactions enter in, and they tend to glucuronidate the molecule for elimination. So that was just if anyone is into biochemistry. There are groups or families of these P450 enzyme types. So 3A4 is the most common site, and it really involves itself with a lot of drug interactions. We're going to talk a little bit about that. But there's other ones too, 2D6, 2C9, 2C19, and we're going to come back to them because it's the other families of enzymes that have a pharmacogenomic variation and a clinical application. Yeah, go ahead. Okay. Okay. That is proactive safety. So liver function tests, ALT, AST, ALK, phos, are indicators that hepatocytes, liver cells, might be in trouble, but they're not real exact. So sometimes they use 3 times the upper limit of the 3 times the elevation of normal as a hallmark. But if the liver is really in distress, you'll see levels that are typically 40 and 50 go up to 300 or 400. So do you remember what your cutoff is? It's simply because a lot of drugs can cause hepatotoxicities, and they want to keep people safe. So that's protecting your research subject. So I'm going to guess periodically you're measuring liver function tests? Yeah. And how elevated? Do you remember? Yeah, okay, so ×2 is almost expected, and that could be a little bit, we'll talk about it. Sometimes a drug will cause more enzyme to be produced, and you'll get more transferase produced. And it's not necessarily a hallmark of toxicity, however, because you want to protect your research subject, and drugs can cause liver toxicities, that's where that's coming from. So they stop the drug, yeah. They'll probably come full circle and try to study that. But it makes sense because, do you remember if your drug used P450 enzymes? Yeah, that's safety. That's safety. Gosh, all right, so I'll take a little side step, and then we'll take a break. So you know about the new, well, they're not so new anymore, the oral anticoagulant drugs. The first one actually came out as much as 15 years ago, and it was on the market in Europe. And it did its job, it anticoagulated people, but they had deaths from liver toxicity. So when liver toxicity is bad, it's very bad. Another really popular drug, some of the diabetes medicines, pioglitazone, the original drug in that class got yanked from the market because few people died of liver toxicities. And I have to say, natural products, so I wonder I'm guessing in your study that they follow the patient's liver function tests, because herbal products have caused, yeah. Oh, yeah. Yeah, sorry. Yeah. So, yeah.
Absolutely.
Yeah. Yeah. Because herbal products, who knows? Plant products can cause liver toxicities. Okay. So should we take a break?
Sure.
Take a little break. Yeah.
Yeah. Back, 2.
Yeah.
5 minutes of the restroom.